Mushroom Wonderland.
Hey, what's going on? My name is Aaron Hilliard, Mushroom Wonderland. Today I have with me a pretty famous rock star mycologist, Alan Rockefeller. And then we have the Fungi Night. Guys, we're here in Washington State in November. So thanks for joining Mushroom Wonderland. Hit like, hit subscribe. Thanks.
I got into mushrooms about 20 years ago. I went mushroom hunting on Christmas Day in California, and there was mushrooms everywhere, and I figured there must be people that knew which ones were common, rare, and edible, poisonous, hallucinogenic, but that was back before many people were on the internet, so it was difficult to get information at first. So I was just editing Wikipedia a lot, taking a lot of pictures, and just kind of got more and more into them. I used to do computer security, so I was breaking into computers for a living. And I used to work for corporations and governments and stuff like that. But about 10 years ago, I quit all my office jobs, and I've just been mushroom hunting full-time for about 10 years now. So now I'm the chief mycologist for a company called Mimosa Therapeutics, and it's a small psychedelics company. And I pretty much just go mushroom hunting all the time, and then also study mushrooms. So I bring a lot of stuff back into the lab, DNA sequencing, microscopy, get really good pictures of it, and then a lot of people send me mushrooms in the mail that I study. So I've been doing that for about a year, and I like it quite a bit.
Where are you based? Oakland?
Oakland, yeah.
Cool.
So science can be done by citizens, and especially with like mycology, it's like really useful because there's not too many people paid to study mushrooms, especially wild mushrooms. There's not much money in like taxonomy, so the real scientists go where the real money is, like plant pathology, which is super boring, but a lot of people want to pay for it. But just people can make real contributions to science by taking good pictures of what they find out there, and especially doing DNA sequencing and uploading the DNA sequences to GenBank. But you know, really any kind of careful nature observation can be considered citizen science. You can tell Tubaria from Galerina because the Galerina has a more interesting stem texture.
Right.
They have like all that kind of flacos,
Fuzz,
kind of like chevrons on the stem, and these have more of a smooth stem. These are a little floppier.
Are those edible?
I call these the edible lookalike of the deadly Galerina.
Oh, okay.
Nobody really eats them, but they look enough like cyans that if they were poisonous, people would be complaining about them. Sequence them, we got like 10 or 15 collections sequenced now, and there's definitely a couple of them. But somebody sent me a Tubaria that stains blue the other day, and it's I tested it with the Miraculous kit, and it had psilocybin in it, and sequenced that, and it did come out in Tubaria, so it's interesting that Tubaria can have psilocybin too.
Hey, what kind of mushrooms do we got growing right here?
These are Pluteus, and we got a couple species on the west coast that look like this. One of them is Pluteus cervinus, which is more common in the spring and summer, and then Pluteus exilis is more common in the fall and winter. So I'm going to guess Pluteus exilis for these, but they're really hard to tell apart. Microscopic features of both of them overlap, so you can't just like throw them in a scope and tell for sure which one you have. So you really got to sequence them to figure out which one you have. And I've sequenced a few, and yeah, the sequences from like the early season do come out as exilis, and the sequences from late season do come out as cervinus, and they're pretty different in the sequences. So it's possible you'd be able to tell them apart just by look or taste or texture.
So these are what are known as deer mushrooms?
Yeah, exactly.
Okay. And these are really hardy to compare to the ones I see growing off of a log in the forest.
Well, you know, these wood chips are kind of like concentrated food for the mushrooms, so you know, they're going to have a lot more substrate to be eating, and so I think that's why they're getting so much bigger than you would see on a log in the forest. With all the Pluteus around here, it's tempting to set them up and do a photo, but then again, we should probably go to more interesting places. Anyway, but I'll just do a quick mynet observation. I have a camera in my backpack,
okay,
but you know, these are not perfect specimens, so it's not really worth pulling out a camera for them.
Gotcha.
But I take the pictures with the iNaturalist app, and that way I don't have to process the photos and upload them at the end of the day. I just
plug them right in.
Just plug them right in, yeah.
Cool. Do you use Mushroom Observer or put stuff on there often?
Yeah, what I do is I use I process the photos off my camera and put the really good photos on Mushroom Observer, and then the cell phone photos go directly to iNaturalist, and then every once in a while I will run this script that uploads all my Mushroom Observer observations to iNaturalist. I'll run it like once a month or so. That works pretty well. You know, the iNaturalist reduces all the photos to about 3 megapixels, so for cell phones it doesn't really matter much, but I can put the full 45 megapixel versions on Mushroom Observer and it saves them forever, so I can keep the high resolution ones there.
Yeah, I've seen your name all over Mushroom Observer. I put a couple mushrooms, the Amanita Pholiota, I think these ones I put on there. Out in Tacoma, right at Wright Park, there's a nice patch of death caps that grow right there, and I don't know, I got this interest in mushrooms that are that deadly. It's just interesting how they grow.
It's like in the UK they have this garden that has nothing but poisonous plants.
I've seen that. That is awesome. So cool.
I want to see that sometime. This is Coprinopsis lagopus, and you can tell just because the cap is super thin and it has this vellum on the top is kind of like fibrous. So like little hairs, cool little mushrooms because they come up so fast. You know, they'll only last like a day.
Are these related to shaggy manes?
No, shaggy manes are in the Agaricacea family, so the shaggy manes are related to like a portobello, whereas these are in Satharellacea, so these are related to Satharella. So totally different family. They just happen to look similar, like convergent evolution, but they're not closely related. This was the first mushroom to be renamed. So Coprinus was described based on Coprinus comatus. That's the type species of Coprinus.
Yeah.
And so those turned out to be closely related to Agaricus. So all these other ones, Coprinopsis, Coprinellus, they're not closely related to Agaricus, so he kicked him out of Coprinus. And now what's left in Coprinus is very tiny percentage of what used to be in Coprinus.
It does have pink gills in there, so that looks kind of like an Agaricus, huh? Their gills look pretty pink.
Yeah, that's true. They do go pink for a while.
What are we looking at here?
So Gymnopilus thiersii was described from California in the Bay Area. So that's a West Coast name for this. Gymnopilus luteifolius was described growing on birch in New York. That's an East Coast name. And so probably one of the two, but on the West Coast we also have a couple other red-capped Gymnopilus. You have Gymnopilus dilepis, and that one's a little smaller, and that one has Chilocystidia that's not capitate. So these have capitate Chilocystidia, which means that all the cells on the edges of the gills have a little ball on top of the cell. Whereas dilepis, the cell is just ventricose. It's just like a bottle shape. It doesn't have a ball on top.
Okay.
So if we throw these on the scope and they don't have the ball on top of the Chilocystidia, I call it dilepis, but I think they will because these are pretty robust. So you can call it luteifolius or you can call it thiersii, depending on whether you think the East and West Coast ones are different.
Okay. And these are known as an active species, or that's still kind of to be discovered?
Oh no, definitely active. You can tell because they stain blue a little bit. Often, like in the rain spots on the cap, you'll see some blue, or at the stem bases. It's tempting to get a photo of them just like that. I think I will, just because they're pretty nice, coming out like that.
I might do that too.
Yeah, I should.
Yeah, that's beautiful.
Definitely have kind of a cucumber smell similar to Agrocybe or Psilocybe. Both on the stem and the cap staining blue. So that bluing is the psilocin polymerizing. And so psilocin forms these long chains. And there's a few different polymers that it forms, but they're all blue, and there's a polymerase enzyme in here that polymerizes the psilocin. But the enzyme requires water to work, so if people are trying to store these mushrooms for longer periods of time, if they keep them extremely dry, then it cannot polymerize. I think what I'll do is really carefully check the microscopy on both of them, see if I can find any differences microscopically. And if I can find consistent microscopic differences, then I'll call this thiersii, and I'll call the others Luteifolius, and if I can't find any difference, I'll just be like, yeah, there's just a little bit of difference in the base pair, in the sequence from east to west, but it's really the same thing. Never went to college for anything. I I always thought I would go to college, but just started breaking into computers in high school, and got a job right out of high school breaking into computers. And I think anything you can learn in college, you can learn on the internet these days if you have sufficient motivation. Not everybody does, so college is definitely the right place for some people. But for people that are really into stuff, you can learn everything. It just really depends on what you want to do. If you want to be like a researcher at a university, you definitely need a degree. But if you want to work for like a small company or work for yourself, nobody really cares if you have a degree or not.
How many new species do you think you've discovered?
Oh, it really depends on how you define discovered. I think we've probably seen some new species today. I think on a good day we'll discover 50 new species, but there's, it's easy to find new species because there's so many that just haven't been published. It's a lot of work to actually write them up and do a good job of writing them up. So I think I've published maybe 6 or something like that. I don't really keep track, but as far as like discovering and saying like, oh, this is a new species, probably about 200. And of those, probably like 5 or 10 of them I'm actually working on, maybe 20. So like when I see it, I'll take real good pictures of it, save the collection. So when I want to publish something on it, I got a bunch of different collections, a bunch of different DNA sequences, and lots of microscopy from all the different collections. So I know what the true variation is between the different species. There's a lot of mushrooms that look the same, but they're actually different. And that can be due to convergent evolution, like 2 mushrooms that are not closely related at all just happen to find that the same sort of ecological factors are impacting them, so they end up looking the same. So a microscope is really good for figuring that stuff out, because the things will look the same in person, but under the microscope they're super different. Or different mushrooms could look very similar because they are closely related. So if you want to see how different they are, DNA sequencing and microscopy is really useful. We've only been doing sequencing for about 20 years, so there's a lot of species that just haven't been sequenced yet. So to figure those out, you need to do the microscopy. So I find that the microscopy is a much better use of time once I have a DNA sequence. So I save everything I photograph, and then I sequence it all with the ITS gene. If ITS doesn't answer my question, then I'll sequence some more genes. And if I don't get any good, confident blast matches, once I blast my sequence, if I'm not positive what it is, then I'll use the microscopy. So people have been doing microscopy for a couple hundred years, so all the literature is based on microscopy. So the DNA sequencing is very new, but you can compare with all the old literature with the microscopic features.
What's your favorite mushroom to eat as culinary mushroom?
Amanita villosa by far.
Amanita villosa.
Yeah, villosa. It's endemic to California. It grows a little bit in Oregon and Baja California, but it's one of those grisettes. It's a pink grisette. It's super delicious. It's kind of tastes like shellfish, a little bit like scallops or something.
Where's your favorite place in the world to forage mushrooms?
I like Mexico a lot, but really wherever it's raining. If the season's good in Michigan, it's awesome in Michigan. California is really good when it rains, both northern, central California where I am in Oakland, southern California. Really, wherever it's been raining the most. You can go to like water.weather.gov/precip, and there's a map of shows where it's been raining the most in the past few days or weeks, up to a month. So you can go on there and see where the rains have been, and then just go to the place that's had like the most rain in the past 3 weeks. And temperatures and wind have a lot to do with it too. If there's a dry wind that comes along, like everything dries out in like 2 days.
The nibble and spit test, is that safe with all mushrooms?
Yeah. You can taste any mushroom. You
Death cap?
You got to eat quite a bit. Here's my box from yesterday. So this is all from Mount Rainier.
Cool, little Stropharia.
This one's really cool. This like little lichen with the black head. We put this in the microscope the other night. It's beautiful.
Awesome.
Yeah, this is like...
So that's not a mushroom though.
Well, it's a lichen. I think it's called Piloderma acicula. And so it's a collaboration between fungi, algae, and yeast. So all 3 organisms growing together.
Wow.
Yeah. And then This one is a slime mold, and it's a fluorescent slime mold. So it looked really cool in the black light.
Cool.
Here we got some Atheniella adonis. So this was kicked out of Mycena a few years back.
Tiny.
This is really cool. It's a mucronella, so it looks like little tiny icicles just growing on an old log. And there's thousands of them, so I got cool macro photos of that.
Be tough to spot that when it's frosty out. Huh?
Yeah, this bird's nest I like. I don't know which one it is. I'm just gonna hang on to it till it opens up. It's probably one of the common ones, but I got really nice macro photos of it, so
cool.
That would be good. This one is Psilocybe pelliculosa.
Oh wow.
So that's a hallucinogenic one growing on a Douglas fir cone. And if you look way up in there, I don't know if it shows in the video, but you can see a few purple spores up in the cap there, and that's how you know that it's not a Mycena or a Gallerina. And here is Stropharia ambigua. These are cool because they have these crystals at the base of the stem. And I think like with a polarized light, you can make the crystals have like all these crazy colors.
Oh, cool. Those are pretty common around here. I think there's some right behind us we can go look at.
Yeah.
So you're going to take some of those?
So I'll take some of these. I will spore print the ones that I can, and I'll sequence them. I'll sequence ITS, LSU, TEF1, and a couple more obscure genes. Because I got that material from New York, so I'll sequence a bunch of different genes and see if I can find any differences between this one and the New York material, so we can figure out if these should be called Gymnopilus thirsii or Gymnopilus luteifolius. The mushrooms are the fruiting bodies, and so when it starts to run out of food, then it starts making mushrooms. It's kind of like, oh man, I got to get out of here, so it makes mushrooms so the spores can move, but they come back as soon as it rains. So really, this patch is going to disappear when all these wood chips get consumed or when a contamination comes, but it won't disappear any faster if you pick every single one. But if you pick every single one, there won't be spores going into the air, but 99.99% of the spores fall right here where they won't do any good. So if you really want to spread them around, you can just take some of the mycelium. Like this is all Gymnopilus luteifolius mycelium. There's probably 50 pounds of it here, and then just bury it in other patches.
Right.
Like if you dig down here, you don't see that mycelium here, so that'll probably fruit next year. But the best thing to do is take the mushrooms, pull them out of the ground, take these stem bases and put them in the pile of wood chips that's 100 meters away. And that way, in 5 or 10 years, this patch will be gone, but 100 meters later, 100 meters out there, it'll still be going from the stem bases. So that's definitely the best thing to do. Just moving it around will help a lot, especially since it's going to rain this afternoon. It's a pretty good time to move this mycelium around. And then, so I just take a whole bunch of it here. Wow, it's really warm in there from the composting. So I spread all this around, and then I take fresh wood chips. And these are not fresh, but ideally I would find fresh wood chips. And see this place where I removed these, it's got all the Gymnopilus mycelium on the edges. So by taking fresh wood chips and putting it here, now all these fresh wood chips will get colonized. So you're not really removing anything if you take fresh wood chips and put it where you took the old mycelium from.
Huh. Some pretty healthy mycelium, it looks like.
Yeah, it's doing really good here. See look at the gill color? Look how they're not turning brown.
Yeah.
So these are Pholiota spumosa.
Oh.
And Pholiota spumosa look awesome in the black light. If I'm lucky, I have a black light here. If not, it's in my car. Don't see it here. Maybe it'll be down here. Looks like I do not have any black lights on me right now. But these are spectacular at night, the black light. I got a couple really good black lights in the car, so maybe we can do something with black lights, but maybe it'll be really rainy. I don't know. You can see there's some spores. If this was a sulfur tuft, the spores would be dark purple brown, just like Psilocybe.
Okay.
But you can see the spores are medium brown, like kind of like medium milk chocolate brown. I call it Pholiota brown. Also, the cap is real viscid on this. And Hypholoma have a little bit of a drier cap. So they look a lot like sulfur tufts, but they're probably actually edible.
Oh. I would never think to eat those, but there is.
Yeah, people don't eat them, but they're probably good.
Well, so what do we got there? This one's super interesting looking. Is this just a really bright colored Stropharia?
Yeah, Stropharia ambigua.
Okay, so it's one of these.
Yeah.
Just young.
These, yeah. That's pretty unusual to see in wood chips. I don't think I've ever seen Xylaria hypoxylina in a wood chip pile before. But it has these asexual spores on the top, these white ones, and then later when it gets more mature, it'll make sexual spores that inside perithecia, and that'll be kind of like dark olive brown spores. This is Peziza.
Peziza.
By Peziza varia. And Pezizas are actually edible. They're ascomycetes. They make spores in asci, kind of like morels, big old cup fungi. Peziza are really beautiful in the microscope if you mount them in Miller's reagent, because the asci have amyloid apices, so like the tip of each ascus it turns bright blue. They're super cool in the microscope. A lot of stuff that looks really boring looks awesome in the microscope. Sure. Here's this is cool. This is Entoloma vindobonense, or I think Nolena concentrica might be a good name for it, but they kind of grow in grassy areas and stuff.
I think about some type of tomato species.
Yeah, this looks like these gills are pretty dark. I think they're a little too dark for Paniolus phonosecii. So this is probably Paniolus olivaceus if it's got roughened spores, or if it's got smooth spores, then Paniolus fimicola. Could also be Paniolus acuminatus. Definitely one of those. It's pretty tempting to study these. No one's studying Paniolus. I think I'll just make a quick INAT observation, throw them on my dashboard, and sequence them and do some microscopy later on.
So these are all saprotrophic mushrooms, right?
Yeah, all of these.
This is actually a rare like redwood growing here in Port Orchard, Washington, which doesn't happen often, but from what I understand, there's no mushrooms that have a mycorrhizal connection with the redwood trees, huh?
That's true. They might have some endophytes, but those never make mushrooms.
So it's kind of like a cedar, huh? Like
all the mushrooms with redwood are saprotrophic for sure. Yeah, it is like cedar.
I never find anything.
Most trees don't really make mycorrhizal mushrooms. It's just, you know, the conifers, the ericaceae, the fagaceae, so like the oaks and the birches, alders, and, you know, some stuff, but the vast majority of trees don't have any ectomycorrhizal associations.
So I've heard it said that all mushrooms are saprotrophic until they find a tree.
It's got to be true, because the spore's got to germinate somehow, you know. It's not like the spore falls right on the root.
So there could be chanterelle mycelium just growing in the woods but not connecting with the tree, and
exactly.
you just won't see a fruit until it connects with the tree. Is that how it works?
Yeah, and you can throw these things on agar and they'll grow. They're not very happy, but I got Amanita muscaria, pantherina, all on growing on agar.
But they're gonna need a tree to make a piece of fruiting body, huh?
Yeah.
So that's why morels grow after a forest fire, right? They're mycorrhizal. They're trying to get out of there.
Well,
And
I don't think they've actually found any morels on root tips, so they're not ectomycorrhizal like a chanterelle is, but they do care very much about the trees, and they do grow after fires, some of the species, so it's complicated.
oh, okay. I guess I got the idea that they were growing with those trees. The trees caught on fire, and they thought we better get out of Dodge, so they spread their spores.
That's what I think the morels fruit after a fire because the trees die, because when there's just a dead tree from like a pine beetle or something, I also see a lot of morels fruiting around them. So I think
they're like, it's time to fruit, it's time to move on, find some healthy.
Yeah, and so, you know, sometimes I think maybe maybe they don't even care at all about the fire. All they care about is the tree dying, and it just happens that, you know, fires cause the trees to die in stress, and then the mushrooms freak out, and they're like, okay, time to fruit.
Time to move, huh?
Yeah.
Yeah. Well, it sounds like my theory might be onto something. So.
Yeah. So there's a couple of species that look like this, Clitocybe nuda. This one smells like orange juice. And then there's another one that's more common with conifers, and it smells kind of unpleasant. Both of them are bluets.
Bluets, good edible, huh?
I don't know. They have a really strong flavor. Some people like them, but most people don't really eat them very much.
Right.
I made a curry once and it overpowered my curry.
Wow.
Yeah.
All right. Well, worth trying anyways.
Here's the gill color.
Oh, Paniolus?
No, this one's got rusty brown spores. And if you look at the stem, you probably can't see it in the video, but if you look real close, the stem apex has these longitudinal striations. So this is Conocybe.
Conocybe.
And a lot of species of Conocybe, you kind of need a microscope to tell them apart, but if we're not going to use a microscope, we'll call it Conocybe tenera.
Okay.
But if we actually scoped it, the chances of it being tenera are very low.
So maybe something nobody's ever described.
Yeah, it could be, or it could, could be in some obscure literature somewhere. We just have to measure the spore size and look at the collocystidia, fluorescent, and best thing to do is sequence it and see what it matches in GenBank. But a lot of Conocypes haven't been sequenced yet, so it's definitely a good one to study if you like studying obscure stuff. I study them when I find a real good collection, so I can get real nice photos. I sequenced one from Point Reyes a couple weeks ago and didn't have any matches in GenBank at all.
So if there's a mushroom that doesn't match anything,
So then you need to use a microscope to figure out
if anybody,
in the literature that's
okay. And if not, then you just say, this is called the Rockefeller.
Yeah, well, you can't name it after yourself.
Ah, somebody else can.
It's best not to name it after a person, because in 10,000 years they're still using that name. They'll wonder who the person is.
Yeah, that's a good
So
point.
it's best to name it after a distinct characteristic that separates it from its closest relatives.
Oh,
So
that's
as you're
good.
studying it, maybe you'll find something that you notice is a little different about it and name it after that.
Uh-huh.
And this is a Strobilurus.
Strobilurus.
Yeah, it looks kind of like a Mycena, but really closely spaced gills and has the yellow brownish stem.
Wow.
Probably Strobilurus trulisatus, which likes to grow in these Douglas fir cones.
Anyways, we've been picking with Alan Rockefeller. This has been super interesting, so I hope you got some value out of this video and hit subscribe. Thanks. Peace.
Thanks for having me on.
Yeah, totally.